Do you see my slides?
Not yet.
Not yet. Oh,
Not yet.
okay. Sorry. Let's see. Um, do you see anything?
Nope.
Okay, well, Zoom says I'm sharing my
No.
screen. Goodness. Still no luck?
No, do you maybe want to email them to me? Then I can try from.
Yeah, I'll do that. I'm so sorry. I don't know what's going on here. I'll send the.
Sorry.
No. Always something. Okay, I've emailed it and put it in Slack. Again, my apologies. I don't know what's going on with
All right,
students.
I'll check my emails now. Thank you for that. I'm just quickly going to check if you can see my screen. I'll start sharing now. Is this your presentation?
Yeah, that's it. Thanks so much. If you just click the slideshow button near the top there.
Okay, I'll do that quickly. I just want to let you know that I'll switch devices because then I can't access my laptop if I'm sharing. Then I'll mute myself here and then join on another device.
Right. Okay. Just let me know when I should start.
Okay. You can start once it's in slideshow mode.
Okay. Okay, great. So yeah, so sorry about that. So yeah, my name's Glenn Hickey. I work for Santa Cruz, and I'm going to talk to you about constructing and using pangenome graphs. Next slide, please. Next slide. Oh, thanks. So an overview of my talk. Yeah, so mostly I want to introduce to you two tools, VG and Minigraph-Cactus, they use for pangenome graphs. To give you some context and motivation to hopefully use them and make progress with them in your Ref Graph hackathon. So an outline of my talk, I'm going to talk at the very beginning with some basic terminology, just to make sure you've got it. It might be redundant with some other talks. And then go chronologically through variation-based pangenomes, how we used to make them with short reads, and assembly-based pangenomes that we'll focus on, which has become possible with long reads, and then with some of the applications that we've used our pangenome reference graphs for. Next slide. So the background, just to talk about pangenomes and graphs. Basically, this will just be a review for most of you. Next slide. So according to Wikipedia, anyway, the origin of the term pangenome is from this 2005 paper where they sequence 8 Streptococcus agalatiae, if I was pronouncing that wrong, genomes. Each genome is about 2 megabases, I think, and a couple thousand genes. Anyway, they realized right away things that we're still talking about now is that if you look at any one of these strains, it's diverse enough from all the others that you can't use it as a single reference. There are too many genes that are in the other ones that aren't in it for it to be useful to be a reference alone. So they put a cohort of 8 together and called it a pangenome and analyzed them within context. And they defined some terminology like the core genome, which is genes that are in all the genomes in their cohort or in their pangenome. Dispensable genome, which are genes that are, private to one strain or to a subset of strains. And I always like to put the abstract here because, this is from almost 20 years ago, and it's basically the same sort of thing that, anyway, I put in all my abstracts today. Genome sequencing is getting less expensive, and one genome doesn't reflect the variability, et cetera, in this case, pathogenesis in bacteria. Next slide. So their pangenome was essentially a set of genes, and you can make a matrix that shows the presence and absence of each gene for each sample. And so their pangenome boils to, you put the samples in the row, the strains in this case, the genes in the columns, and one if that sample has a gene and one if not. So the columns where there's ones all the way down, like B and D. Next slide. That would be the core genome in this case. So that's how pangenomics started, at least with respect to this paper. Next slide. And you can also just look at the size of this common core genome with respect to the number of genomes. And they extrapolated that their 8 genomes would be sufficient to find all the core genomes of this species. I'm not sure if that was true or not, if they sequenced more. And this sort of is related to what Benedict said, in that you don't usually need too many genomes to find most of the common variation. But depending on the species, you could add many more private variants as you add more individuals. And that comes into play in humans as well. Okay, next slide. So yeah, this was all well and good for prokaryotes, and it was immediately obvious these strains are diverse enough that you need a pangenome to do anything. But there's no reason that we can't apply this to other organisms. So back then, the limitation was just access to sequencing data. There was no human pangenome back then. But the technology's evolved, and we've had more and more sequencing. So for eukaryotes, if you made one of those matrices, for people with the genes and the samples, most of the genes would be the same. Now, there's a few differences between individuals, but there's not a ton of signal. But if you have a whole genome sequence, there's variations within the genes, there's variations without of the genes. That's quite interesting. So we have to move, when you move away from bacteria, you have to look at pangenomes of the sequence itself, not just sets of genes. And you could argue that the 1,000 Genomes Project, which cataloged a bunch of human variation, they didn't use the word pangenome, but it was definitely in the spirit of pangenome. Okay, next slide. So to talk about pangenomes of sequences, the most natural way most people agree on is to use graphs. And so just to review again what a sequence graph is, if I have a set of genomes here, G1, 2, 3, and 4, each one's a DNA sequence. When I make a graph out of it, I kind of take all the common bits of sequence and I switch them into one node. So each genome here begins with TGA, CCT, AGA. That becomes a node in my graph. And then I have a variant there, a C to A SNP. So I make a node for each one, et cetera, and et cetera. So the parts through the graph, that's just one node that's common to all the genomes, kind of a little bit like those core genes we saw before, except now we're dealing with sequence in its context. Each sample, yeah, this is a good slide, thanks. Each genome in the, oh, sorry, back one, please. Right. So each genome can be threaded through the graph. So if I take G3, this is its path through the graph. So my sequence graph in terms of pangenomes is three things. It's the nodes of the graph, the edges that join the nodes, and each genome that goes into the graph is a path in the graph. So usually when I talk about a pangenome reference graph or a sequence graph, that's what I mean, nodes, edges, and a path for each genome. The next slide. So if you go back to the 1000 Genomes project, I'm sure a lot of you are aware of VCF files, variant call files. VCF is the basically the text you see here in the middle of my slide. It gives you a set of variants and positions. So the first line of my VCF says that position 10 of chromosome 1, the C in my reference genome changes to an A in this sample, et cetera, et cetera. So the VCF files we've been dealing with from the 1000 Genomes project, for example, they're very much analogous to pangenome reference graphs. You could turn a VCF, you could think of a VCF as a reference graph, where you have the reference genome, which could be GRCH38 or GRCH37, whatever. And then you have variants off that reference genome. So that's what the picture is showing before. You know, highlighted in black is the reference genome. We'll pretend it's GRCH38. And then my VCF shows two variants on top of that, a C to A substitution and an insertion of two T's at position 21. Next slide. So that'll lead us into the variation-based pangenomes. So this is going from short reads to variant calls, like you have in the 1000 genomes. So variant calls in a VCF and make a pangenome from that. So next slide. Next slide, please. Okay, yeah. So that's it. So there are two applications, just a little bit of a digression here. If you have a pangenome, you could use it for two broad categories of analyses. You could use it to study variation inside your pangenome. So your pangenome is made with a set of samples, and your pangenome itself shows you how all the samples relate to one another. So that, I think, is like what the 1000 Genomes project is. So they started with a reference genome. They sequenced short reads for thousands of different individuals. They called variants against the reference for each sample. And then they listed all those variants in a table, like a VCF, or they could even make a matrix like that other one I showed you, presence absence matrix for GWAS. And that framework is great for looking at variation within your pangenome. But if you want to use your pangenome itself as a reference to reduce reference bias and take advantage of the variation you learned for the samples in your pangenome and use that diversity for samples out of your pangenome, then your list of variants falls short. You're better off you needed some kind of graph, reference structure you could work with. And that's where pangenome reference graphs come into play. I think the really major step in that was the original VG paper from Eric Guarracino, who took the 1000 Genomes VCF and turned it into a literal pangenome that you could do something with. Next slide. So this starts with the VG. So VG is a software suite, a tool set that we maintain to this day, and it started with Eric Guarracino about 5 years ago. And so VG could take a VCF file and convert it into a sequence graph. And it provides you a whole set of tools to index and manipulate and even map reads to the sequence graph. So this is a figure from the original VG paper. And it shows you for heterozygous variants across the genome, the map reads to it. And you would extend y-axis is the y-axis, excuse me, is a fraction of reads covering the alternate allele at these heterozygous sites. So you expect basically that to be 50%. You'd want 50% on your reference allele, 50% on your alternate allele, barring some, differences in your sequencing coverage. And that's what you get in VG across the board. The x-axis is a function of indel sizes. So big deletions on the left, big insertions on the right. When you compare that to BWA-MEM, once you get into bigger or smaller insertions or deletions, you're seeing far more reads on the reference allele than the alternate allele at these sites. And so that is a snapshot of reference bias. That means the reference genome you're using, I think it was HG19 in this case, is pulling in reads away from these real variants, especially in the case of these medium-sized indels. And the pangenome graph they're able to show rectifies that almost completely. Next slide. So this is a diagram that comes up showing the VG toolkit. I won't go into too much detail, save for the fact that the idea is that you have a bunch of variant calls, usually from short read data for different samples, combine those into a pangenome graph, and then you're able to use that pangenome graph to map like this n plus first sample on the top right. And you're leveraging the variation you know about from samples 1 to n to improve your read mapping for the n plus first. And VG also contains some tools for genotyping and variant calling and visualization and extraction, et cetera, et cetera. Next slide. Next slide. Next slide, please. Sorry. Okay, so start to moving on from VG. So VG can work with any sequence graph, right? A sequence graph is just nodes, edges, and paths. And so VG could deal with any sequence graph like that. But in practice, you're limited to constructing graphs based on these variants that were called against the reference. And this started to become an issue for complex regions, especially in terms of structural variation. Because, well, if you've ever dealt with SVs and VCFs, the format isn't suitable for it. But there's also things that, if you call a bunch of different samples against the same reference, you don't really get notions of nested variation. So here at the bottom, I have a simple case that you can't really do properly in VCF. So I have my chromosome 1 reference at the bottom, and there I have we'll pretend it's a big insertion. And inside my insertion, there's an A to G SNP. So that's a SNP that's nested within inserted sequence that itself isn't in the reference. And there's no good way of doing that, especially as it gets more complicated. If you have like an inversion nested in insertion, et cetera, you can represent it all in your graph like I do here. But you can't really represent that or compute that in VCF to put it into the graph in VG. We know because we tried this paper I'm citing here, we're doing variant calling with VG on structural variants, and we broke our heads making graphs from VCF with that quite a bit. Next slide. So this moves on to the sort of next paradigm that's just starting now, is to skip the variant calls mapped to the reference. And now that we have long reads and great assemblies, we can make a pangenome directly by aligning them. Next slide. So that's it. So this is only very recently now that long reads have gotten accurate enough and available enough to a certain extent. And that we have assembly tools, which I think you'll learn about in the talks tomorrow, like HiFi-ASM and Verco, that is making it much easier to make amazing quality assemblies now, like assemblies that are even better than GRCH38. They're phased. That means you get 2 genomes per each person, a maternal and a paternal one, and they're very contiguous and they're very high quality. And the Human Pangenome project, which you heard about from Ben and Karen yesterday has really been at the forefront of developing these technologies and releasing these genomes. Next slide. So this slide is kind of ancient history now, but the big human draft pangenome paper came out this year with what we call these year one assemblies. And they were made from 1,000 genome samples. And they're using hi-fi reads from PacBio, I think about 30x. And each sample came from a trio. So we had Illumina reads from their parents. So they were assembled with hi-fi ASM, which was able to use the parental reads to phase the assemblies, basically. And so the output of all this, after much deliberation, was 47 high quality diploid phased assemblies. Next slide. I won't belabor this too much, but very high QV 53, so that's very few base errors. Next slide. And yeah, okay, that's it. Next slide. And they're not T to T, but the N50 is, you know, in the tens of megabases. So like I said, this is ancient history. We're now in the process of making the next round of assemblies that will be much, much better than even these. And there'll be more of them too. Next slide. So now we have these assemblies in hand, so we should be able to do better than just what we're doing with the short reads, calling variants on the reference and making that type of pangenome. Ideally, what we'd want to do is take the assemblies and make a pangenome directly from that. So you can see my cartoon pangenome graph there is full of what's a complex region, I forget which one, from a real human pangenome, full of nested variation. You can't do that based on variant calls to the reference. So the idea here is to combine our assemblies directly into a pangenome. The problem is that to do that is more or less equivalent to making a multiple genome alignment of every genome assembly. And that's a classic and very difficult problem in bioinformatics. And there still aren't very many tools to do so. Next slide. Okay, so this is one of the first approaches, still very recent, to make a pangenome from an alignment assemblies is Minigraph. This is from Hang Li. And it does something familiar. Any alignment geeks would know partial order alignment. That's an iterative alignment approach. And that's essentially what Minigraph takes. So if you look at the cartoon on the right, Minigraph starts with a reference genome. That's the red arrow there at the top. And then it adds in the next genome, ASM2 it's called. That ASM2 has maybe a big insertion bubble. That's fine. And then it puts that into a graph. So you got the red line with the blue bubble. And it indexes that graph. And then it adds in a third genome, ASM3, which kind of modifies that bubble to add the green arrow, et cetera, et cetera, et cetera. So Minigraph is an iterative procedure. It starts with a reference genome, but it's able to handle these nested variants because each time it adds a new genome, the genome after that has a notion of that. It's not all aligning to the reference. It's building up the graph. And Minigraph is nice in that it's very, well, relatively fast and easy to use. But it has some limitations. It could only represent variants of a certain size. In this case, usually about 50 to 100 base pairs. So only structural variants in Minigraph. And that's a problem if you're interested in small variants, if you're interested in these nested variants, even if you're interested in building pangenome graphs, as I've defined them before, as nodes, edges, and paths. If I don't have every variant in my graph, I can't represent my sample as a path in the graph because I don't have all the sequence from any one sample in my graph. Next slide. Yeah, so this finally brings us to Minigraph-Cactus. So Minigraph-Cactus, the idea was to take the SVs from Minigraph and then refine them to include all the small variants using Cactus. Cactus is an existing genome aligner for aligning genomes of different species that Benedict and I worked on years ago. And so we kind of rewrote it a little bit to be able to work with mini graph alignments. I won't bore you with all the details of what went into that, save to give you a little overview of how it works. So if we go to the figure here, you start off with the mini graph as I described before. Mini graph starts with this reference, the blue one here, and then it adds a genome here, an orange one. So this is what an inversion looks like in a sequence graph. If you follow the orange genome in that second picture, you see it kind of goes through that second node backwards. So that's an inversion. And so that's my mini graph. We'll say it's very simple. It has one structural variant inversion in the middle. Then I use Minigraph works as a mapper as well. I take all my genomes and I map them back to that graph. And I get a genome to graph mapping for each one. And that's this middle picture, number 2. And then finally, I feed all those mappings into Cactus. Cactus works by taking a bunch of pairwise mappings, building a graph out of them, and then refining that into a sequence alignment. And so Cactus turns that into a base alignment. So now here I have a path for every one of my genomes through the graph, and you could see how much more complex it is. And this is a real example. I forget from exactly where, but it sort of illustrates, and that will be a point going forward, is that once you add the smaller variants to your pangenome, the graph gets much, much more complex. Next slide. These are some stats from the graph that was published in the HPRC paper. In fact, we did 3 graphs. We did a Minigraph graph, a Minigraph-Cactus graph, and a Pggb graph. And Pggb is what Andrea is going to talk to you about, so I won't go into it here. It has some important differences with Minigraph-Cactus, so that we could discuss after or during the workshop. Anyway, the take home here is that you have far more nodes and edges once you go from Minigraph to Minigraph-Cactus. You go from, let's say, 600,000 edges to 113 million, so thousands more. And the length of the graph, which is the total amount of sequence of the graph, doesn't change very much. You still have GRCH38 plus about 100 megabases of novel sequence. And all this right now takes about 30 hours to produce if you have a cluster. Excuse me. Next slide. This is just an overview of the amount of small variation you get in Minigraph-Cactus. So if you look at all the sites of variation in the graph, and you kind of measure how big they are, that's the x-axis here. So you have structural variance on the right and SNPs on the very left. You could see that, and it's a log scale, that you're getting many, many more SNPs when you add in the small variance. The next slide. Okay, so this is something that everyone talks about when they write pangenome papers, is the amount of variation or the amount of novel sequence they found in their pangenome that wasn't in the linear reference. So if you always start with a reference when you use Minigraph or Minigraph-Cactus. So we started both with the T to T CHM13 and GRCH38. And this plots out the amount of novel sequence in either graph that's not in the chosen reference, and the number of haplotypes that went into the graph that's in it. So there's about 100 and some odd bases that's in at least one haplotype on the left. And then if you go all the way, there's still, especially on the GRCH38 graph, because GRCH38 is so incomplete relative to our new assemblies, there's still a couple of megabases in every sample that's not in GRCH38, just because of holes in GRCH38. So the short story is that we found about in our 44 samples, we found about 100 megabases of sequence that wasn't in the reference across these samples. A lot of it was fairly common. You know, if you look in the middle of my plot, there's about 10 megabases that's in half the samples that's not in the reference. Some of the sequence contains genes. All of this sequence is euchromatic. We're not even dealing with the centromeres. If you add those in, you get gigabases of new sequence. Next slide. Yeah, so you can make plots with your pangenome graph a little bit like the one I showed you for the bacteria yeah, looking at this novel sequence across how many haplotypes support it. I guess this is a different view of the previous slide. You can see there's a bit of a step toward the left hand, and that's just because some of the haplotypes, the columns are by haplotype and not by sample. So some haplotypes have an X chromosome, some haplotypes don't. But it's all to say that you have a couple of megabases shared between everything that is off the reference. And next slide. So yeah, I've mentioned, as I've alluded to before, when we're looking at all this novel sequence, we're not considering the centromeres, and that's because the Minigraph-Cactus and Minigraph leave them unaligned. And because they're unaligned, they're not so interesting in our graphs, so we tend to remove them. Removing them is a bit of a compromise. We're losing real variation, but we're also making a more simple linear graph because of it, and it allows us to align genomes to it a little bit better. So this is a chart of the amount of sequence we've removed from each sample in the graph, and it comes up to about 200 megabases. And it's removed for different reasons, but mostly just because it didn't align well. So the bulk of these 200 megabases is centromeric or other satellites. Next slide. So this is kind of what that process looks for. And if you do the workshop, you'll see you'll get both versions of the graph. If you make Minigraph-Cactus on the left, you'll get the full graph. So in the middle, that's a centromere. And you can see every path goes through its own node. So that's what an unaligned graph looks like. In practice, we normally just clip that out. So that's the right graph. We just cut all the paths where they go through it, and we leave one reference path through it, just so we keep our coordinate system. And that graph on the right, we lose the variation in the centromere. But for the rest of the genome, we're able to effectively map reads and call variants and genotype variants with it. Next slide. Yeah, just some citations. So this year alone, we've made obviously the graphs for the draft pangenome reference. That's the HPRC paper. Then a few weeks later, the Chinese reference genome came, and they made a Minigraph-Cactus graph, excuse me. And then just the other week, there's a preprint for this Arab pangenome reference, and they're also making their own Minigraph-Cactus graph. So more and more of these graphs are coming out in recent pangenome papers, and they're all using them for similar types of analysis like I have and will show you. Next slide. If you go onto the Cactus website, you can find a lot of these graphs, at least all the graphs I've made. So that includes the HPRC, the Human Pangenome Reference Graph. I've done fly, dog, chicken, some other organisms. And you can find all the outputs and all the inputs and all the commands that are used there. So that you can find from the Minigraph-Cactus documentation, the Cactus website. Next slide. Next slide, please. This is just an example of, and again, those of you in the workshop, I hope you'll be looking at this a little more in depth. So the input to Minigraph-Cactus is a set of genome assemblies. So this is an example of a chicken graph. So at the top, you need a sample name and a URL or a local path for the FASTA file. So that's your only input to this. So you make your table of chickens in this case, and then you run it. It took about 6 hours for these. Chicken genomes are, I think, about a gigabase, 30 chromosomes or so, 34. And then it outputs you a ton of files. And then the workshop, if you're interested, goes into detail on each one. Suffice to say that you get different versions of the graph with and without the centromeres, and all sorts of different indexes you can use for querying them with VG and even Odgi as well. Next slide. Okay, so that's Minigraph-Cactus. I'm going to briefly go over some applications here. I don't know how I'm doing on time, by the way, but there's about 5 minutes left. Okay, so next slide. Okay, so one of the first things you could use your graph for that's interesting is visualization. So some of these complex SVs or complex sites, you really need a graph to see. And I hope, and I'm sure Andrea will go into much better detail than this, but there's there are many different, this is still a very active thing. It's hard to visualize pangenome graphs at scale, but what you can do, and you'll see in the workshop, is that you could often pull out regions of the graph and zoom in on them. So I've just put in 4 here. There's others ways to visualize a graph. The top one, A, is using VG. That's one of the oldest ones. It's still an interesting way to look at little regions and paths in the graph. B is Odgi Viz. This is a great one that kind of squishes your graph into almost like a alignment matrix while still showing you some of the structural variation. And that one scales really well. You could do usually whole chromosome graphs with that. And Minigraph-Cactus has an option to spit one of these out for each chromosome. The bottom left is a Bandage NG. That's a great tool as well for visualizing interactively. And then D on the bottom right is another great one called Panicus. That's a new one that could make these beautiful growth curves, much nicer than the ones I showed you before in a variety of different ways. So for people working with pangenomes, and this will come up in the workshop, definitely encourage to use these as much as possible to get a feel for what variants look like in your graph. Next slide. So yeah, the other, another application that we've worked on a lot in our group is read mapping to graphs. So this is a short read mapping in particular. These are 3 ways to do it in VG, starting with VG map from the original paper to newer methods like VG giraffe, which was published more recently. And VG giraffe is kind of the one we focus on now because it's fast and it works well on these assembly-based pangenomes, whereas VG map kind of struggles a little bit. And it's fast enough to be comparable to something like, say, BWA mem. Next slide. Okay, I think, yeah, and it works well with these assembly graphs. Okay, next slide. You could also map long reads to graphs. This is something we haven't worked on so much yet, but there's a good tool called Graph Aligner out there that's very simple and works quite well. We're also very much working on adding long read to support to VG giraffe, but that's not ready yet. But we have shown we can demonstrate measurable improvements in both short read and long read mapping on pangenome graphs with respect to linear references. Next slide. Okay, and I have a slide here saying exactly that. This is the figure from the Minigraph-Cactus paper. Just the top chart is showing that if you, well, it's showing something fairly obvious, but it's good to demonstrate empirically, is that your reads map better to a graph than they do to a linear reference, and that there are fewer edits than your reads. And that makes it easier to use them to call variants and often easier to place them. So if you measure just perfectly aligned reads, you have substantially more on the graph. Then the bottom slide is showing that reads, this is moving to long reads now. You can map long reads, a much higher proportion of them without split read mapping, and add a higher identity to the graph. So in a nutshell, your graph, because it contains more of the variation from your sample in it, it makes the mapping problem a little bit easier because these reads often find a better home on your graph. And this is particularly true, obviously, in places where your sample is much different from the linear reference genome. Next slide. Another big application for the graph is structural variant genotyping. So structural variant genotyping, we use this to refer to using short reads genotype structural variants that are in the graph. So normally short reads aren't great for finding structural variants just because the reads themselves are smaller than the variants you're looking at oftentimes. But if you already have a catalog of structural variants in your graph, then you could use that information to sort of bootstrap your short reads and to tell which variants are in your graph. And we have two approaches, VG and Pangene, that we could explore, both of them in the workshop, that improve structural variant genotyping considerably with short reads. And I think Benedict had a recent slide yesterday showing that with the graph and short reads, we're almost reaching the performance that you get with long reads in terms of structural variants. So just having that information in the graph is really allows you to leverage your short read samples. And short reads are still much, much cheaper than long reads. So most large genomic studies are using short reads. So the graphs are very important to help you to use that information for structural variants. Next slide. And this is, just a measure here that comparing a few different approaches. This is from the old VG call paper. Using the graph, especially on real reads, we were able to improve SV accuracy using a variety of measures. Next slide. And this is, so furthermore, so that previous slide showed how a pangenome graph helps structural variation, but now these new assembly-based graphs, when we compare them to the old variation-based graphs, they further improves SV genotyping. So that's what we're comparing here to the HPRC versus the HCSVC. We call much more variance in the bottom slide, and and our accuracy measures are far, far higher. So the pangenome graph itself helps with variant calling, and then moving to this new paradigm of assembly-based graphs really helps in terms of SV analysis. Next slide. Another interesting thing you could do with pangenome reference graphs is project between the genomes in the graph. So if I have one thing you could do with that is take your mapping to graphs. So if you use VG Giraffe to map your short reads to the graph, you could use this tool called VG Surject to transform your mappings to the graph to the mappings of any genome in the graph. Usually you would do that to GRCH38. So you change your graph alignment to the linear alignment. And there's a value in doing so, because some of the reads that you mapped accurately with your graph that you would have maybe lost completely by starting with the linear reference, you sort of rescue those and can project them to the linear reference. Next slide. And so doing that, you could leverage the pangenome without worrying too much about new formats. So say you want to run a state-of-the-art variant caller like deep variant, which only works on BAMs, on linear references, and you want to look at your variants in VCF, well, you could still use the pangenome. You map your reads to the pangenome, then you project them back to BAM, and then you use that for the variant calling pipeline you're used to. And we've shown that you could actually get quite a big accuracy boost by doing that. That's what we're showing in both slides here. This applies across the genome. It applies especially in the complex medically relevant genes from Genome in a Bottle. And we have a pipeline for that called Giraffe Deep Variant, which is exactly what it is. You map the reads to the graph, the Minigraph-Cactus HPRC graph. You project them to GRCH38 or T to T CHM13. And you feed that into deep variant, which is being trained to understand BAMs coming from Giraffe. And you get state-of-the-art variant calling performance that beats using deep variant with a linear mapper, or even other tools like Dragon and GATK. Next slide. So yeah, a lot of ongoing future work. Some of this was touched on yesterday. We're working on a personal pangenomes that's up and running and in the tutorial. So that's extracting a pangenome graph that's relevant to your sample using short reads and doing that quickly. Because that's an issue with graphs as they become more complicated. And as you add more rare variation, those variants, they could help you, but they could hurt you. If you're genotyping a sample and your graph contains many variants that aren't in that sample, those variants usually aren't helping you. So this is a way of getting around that. Working on the long read giraffes, the centromere alignment is obviously an active thing. We have someone working on that. The goal is to have an option to add the centromere alignments to the graphs one day. Working on off-reference coordinates, the pangenome, I haven't talked about it, but it gives you a coordinate system for all this sequence that doesn't have a home on GRCH38. So to really leverage that, you want to be able to report variants within these off-reference sequences. And so I'm working on making that a little easier to do. And then another thing I'm working on is a comparative pangenomics, because Minigraph-Cactus and Progressive-Cactus, they sort of live in the same software package. One aligns different genomes, genomes of different species. One aligns genomes of the same species. So I like to be able to combine those two alignments. So say align the human pangenome to the rest of the primates, et cetera, et cetera. An interesting ongoing work. Next slide. Okay, just a summary of my talk. So pangenome graphs have two broad categories of uses: to study diversity within the samples, or as a reference for applications such as mapping and SV genotyping. So to leverage the diversity to understand other samples. Minigraph-Cactus is kind of a balancing act between those two things. You want it to have as much diversity within the samples as you can, but not so much that you can't use it as a reference with current tools. In terms of pangenome graphs, we've moved from the old way of making them from variant calls, from short read data on the reference genome, to really just the past couple of years moving to this paradigm of going straight from the assemblies into multiple genome alignments. And we're seeing a lot of improvements across the board by doing so, but many challenges remain, as you'll see when you actually try to use any of these tools. Next slide. Thank you to a bunch of people, especially the Human Pangenome Reference Consortium, Benedict Patton and Heng Li, Jean Malong, my main collaborators, but all the Pggb and BG teams and everyone else. All the tools and documentation is on the Cactus GitHub page. And I've made a tutorial that I'm still working on for the Ref Graph Hackathon that goes into the commands and the explanations for a lot of what I showed you today. So hopefully you could get into that. Next slide. And yeah, I think, that's just a tutorial. Sorry, it's such a long link. It's in the Slack, and anyone who contacts me, I'll give it to them. And yeah, it covers constructing and analyzing and mapping and genotyping on the graphs. And it's all done in Singularity, and hopefully will work on your cluster computer. Thanks, that's it.
Thanks, Glenn, for a lovely presentation. I don't know if the attendees have any questions. Any questions? I guess your presentation was very lovely. It was quite clear for everyone to understand. Are there any questions from the attendees?
I see some hands.
Oh, yes, Hussein, you can ask a question.
Thank you very much for this nice presentation. I wonder how VG works when you have, if is considering the depth of coverage when you construct the graphs. Some of the reads, for the short reads, they have the mutation, and others they don't have the mutation. So let's say you have 50 reads, they have the mutation, and 200 reads, they don't have that mutation. So they have the reference allele. Is that considered when you're constructing the graph or not?
That's a great question. That's usually considered upstream of the graph construction. So usually you take your reads and you make your VCF with a variant caller. And it will be up to the variant caller to decide, is that a reference allele? Is it a heterozygous reference alt allele or homozygous alt allele? And you could use deep variant or GATK or freebase, and they all have models of various complexity to answer exactly that question. Deep variant, for example, is trained on a lot of short read data, and it uses its learned models to try to figure out if you only have 50 reads for the alt and 200 on the reference. It uses its model to figure out if that's a real variant or not. But by the time it gets to VG, VG's kind of lost the notion of that coverage. It only cares if there's a variant or not when you're constructing your graph.
Okay, thank you.
Melissa, you can also ask a question.
Thank you. I just wanted to ask if the workshop materials, like the tutorials, will be made available afterwards for non-participants.
Yeah, I don't know if that's, the tutorial I've written, which comes with data and all the commands, is up there on the public GitHub and will be there forevermore. But I don't know if someone from the workshop will have to answer in the context of the broader workshop. Right?
If I can quickly come in there, thanks for that, Len. So the workshop is a closed workshop for the group in HPRC C Bio, but we thought it would be great to open up the webinar series to everyone within the HPRC consortium so that we can share the knowledge there. But in terms of the materials, we usually do upload our materials onto our website and our YouTube page as well, especially for the recording sessions. So yeah, in that regard, apologies that the workshop is not open to everyone, but we thought it would be a great way to involve people through the webinar series. I hope that answers your question, Melissa.
I can see that Faisal is requesting the tutorial link could be shared on the chat box. I don't know if that is feasible.
Oh yeah, I'll do that right now.
Okay, no problem. I don't know if there are any other questions from the attendees on Glenn's talk. Okay, well, if there are no further questions, I would like to thank you, Glenn, for presenting to us. That was a very lovely presentation, and I hope the attendees enjoyed your presentation. So our next talk will be from Andrea, who will be presenting on building pangenome graphs. I don't know if I've mentioned that correctly. Yeah, okay, cool.
Yes.
Welcome, Andrea.
Hi, can you see the light?
Yes, we can. I can only see a cursor moving around, but not the light.
Okay. Sorry, you said the slides are on. Can you see my cover slide?
Yes, I can see the slides.
Okay, thanks. So hi, everyone. This is the structure of my presentation. I will give a very short introduction on pangenomics because just to repeat very basic concepts, but just a very few. I think it would be helpful to repeat these very basic but important concepts. I will talk about how we build the pangenome graph, and I will show you a few tools how to analyze the graph that we build. So about pangenomics, you heard this several times now. A single reference is not enough to fully represent the genetic diversity of any population, so that we need more and more genomes. And now we are in the era that we can build high quality de novo assembly much more easier than before. And that's why the field is moving from genomics to pangenomics, from a genome to a pangenome. Nowadays, the most general definition of a pangenome is very simply just a set of sequences it started with the bacteria and genes, but now we are more general to say a pangenome is a set of sequences, regardless of the type of the sequences and regardless of on how we represent these sequences. For example, a pangenome on the top right here, that's a pangenome, just a multifasta with the sequences not aligned. This is a pangenome. On the bottom, there is another example. This is the same pangenome, but now here there is a multiple sequence alignment. So we have more information. We have still the same sequences, but here we also know how the sequences relate to each other. So we would like to represent in a structure both the sequences and their relationships. But about pangenomics is not just simply study multiple genomes. It's the way to conduct the analysis that changes. On a genomic approach, what you usually do is to relate all your new samples. You relate the samples against a reference genome. Instead, on a pangenomic approach, you relate all the samples against all the other samples. There is no special guests in the comparison. You relate everything against everything. This is important to reduce what is called the reference bias. The reference bias happens in a genomic approach where, for example, you have your reference here. You have your 4 samples. Sample D has a region that is particularly different with respect to the reference. And when you will map, for example, short reads against the reference, you will tend to lose the reads carrying these variations. The aligner will say they are errors, so you will tend to not map those reads. And that's the reference bias problem. If you compare, you align everything against everything, is more likely to catch also the variation in this region of this sequence. And a structure that we use to represent both the sequences and their relationships is a graph structure, where the regions that are the same, homologous, the same in all the sequences, they will collapse into a single node in the graph, and region of variation lead to branches in the graph. But to be more precise, what we use is the variation graph model. In this graph structure, we represent the sequences as paths through the graph nodes. For example, we have these 2 very simple sequences, 2 very simple genomes that differ only in one position here. This is on the bottom, it's a pangenome variation graph that can represent this pangenome. The first sequence is a path through the nodes 1, 2, and 4. You follow the sequence in the nodes to reconstruct the original sequence. Genome 2 is represented in this pangenome graph as a walk through nodes 1, 3, and 4. That's the structure that we use to represent pangenome. What we would like to want is we have our set of sequences. We want to align these sequences to know how they relate to each other and represent this pangenome in a graph structure to keep both sequences and relationships. I will talk about pangenome graph construction in this second part of the webinar. There are 2 main ways to build pangenome graph. On the left is the progressive approach on which Minigraph and Minigraph-Cactus are based. To the right, there is a null versus all approach that's more general. You compare everything against everything, but it's also harder, more computational, expensive. But this is the approach that we apply in our pangenome graph builder pipeline, Pggb. Pggb consists in 3 main steps: a sequence alignment with a double mesh, graph generation with Sequish, and graph normalization with Odgi. Graph normalization to get a graph that can be used for downstream analysis. So about the first step, I will quickly describe all these 3 steps. In double mesh, there are two main steps. In the first step, we identify homologous regions between the sequences. The problem is that we have two sequences that we want to align, a query sequence and a target sequence. These methods divide the query sequence into pieces and find where these pieces map well enough on the target sequence. I say well enough because this method is heuristic and is alignment free. It does not perform base level alignment. So with this method, we can just identify where pair of sequences share homologies. But we still need to really align these homologous regions. And that's the second step of double mesh. We have this region and we need to align them. The alignment problem can be modeled with a matrix that you fill with respect to a scoring system. You give a score to matches, mismatches, indels. And the alignment problem in this way translates to find the best path through this matrix from the bottom right to the top left. Finding the best path means finding the best alignment with respect to this scoring system. But this approach is quadratic, is very computational expensive. It can be applied in real life sequences. But with a different formulation of this problem with the wavefront alignment algorithm, we can reduce this computational complexity. And this practically translates in, we can find this best path in this matrix by just exploring not all the cells in this matrix, but just a subset of them. So this translates in less computational steps. But what we do in double mesh in Pggb is not to compare. Here in each cell, we compare two characters, two nucleotides. In double mesh, we compare pieces of sequences inside each cells. In this way, it's like having a cell, a matrix with less cells because cells are bigger. That's the intuition here. On the left, there is an animation of what happens. This is called the wavefront. The wavefront explores the matrix to find the best path from the top left to the bottom right. That's to find the best alignment. And here, each pixel, it's a comparison between pieces of sequences, not just two characters. But what we actually do is we have not one wavefront, but two wavefronts that start from the edges of this matrix. And when they meet roughly in the middle, we can split the problems in two subproblems. We can go further recursively. On the right, there is an animation of what happens. These two wavefronts meet and meet and meet again recursively. And this allows to align sequences by keeping memory usage very low. So this is how we align the sequences. Now that we have the alignments, we can really build a pangenome graph with seqwish. With seqwish, what we have, it takes the sequences and the alignments. These dashed lines represent the alignments. For example, these three Gs aligns to each other. And seqwish essentially takes this information and it collapsed characters that align into single nodes. And this way we can build our raw pangenome graph. I say raw because in the process we apply a few filters to prevent possible errors that can affect badly graph generation. So we need still to normalize this graph to be able to use it for downstream analysis. That's what we do in the third step of Pggb with smooth-xg. To explain how we normalize the graph, I have to introduce a possible way to visualize pangenome graphs. For example, we have these two sequences, a pangenome graph that can represent these two sequences. If we put the graph nodes in one dimension, one axis from left to right, we can represent these two sequences in this way. Genome 1 is represented as a colored line that covers nodes 1, 2, we skip node 3 or 4. These black lines represent the edges, so the graph topology. Genome 2 is represented as a colored line that goes through nodes 1, skip node 2, so node 1, 3, and 4. That's a very simple visualization that can scale on big graphs. So if we apply this kind of visualization on a real pangenome graph, that's what happened in the graph normalization. We apply a graph sorting algorithm on the sequence graph, and then we divide this graph in pieces, and we apply a multiple sequence alignment locally inside each piece to normalize the alignment and then the graphs. Okay, and this is the whole workflow for Pggb. So we start with a single FASTA file with all the sequences of the pangenome. We perform the alignments, the graph generation, multiple round of normalization, another step in normalization, and then we can produce the VCF file that represents the variance represented in the assembly. Here we use VG, a graph in Odgi format that we use for another suite of tools that I will show you quickly. And we generate also VCF statistics and multiple visualization of the graph, static visualizations. This pipeline is also available on the Nextflow if someone interested in this kind of workflow managers. Okay, so this is how we build pangenome graphs, but I would like to show you also a few tools to work this with this pangenome graph. What we developed? We developed a suite of tools in Odgi that provides users a set of tools to analyze, to visualize, to explore pangenome graphs. For example, it's really important, also Glenn said, to visualize, to graph, to understand what's happening if pangenome graph generation is going correctly, if you get what you expect. This is a visualization that we can generate with Odgi, a static visualization in 1D. So also here you have to think that all graph nodes are in one dimension from left to right. In this particular modalities, each path has its own color, but here colors have no particular meaning, but we can give a meaning to the colors. And we can color, for example, the path by position. Here, there is a gradient that goes from gray to black with respect to the position and the path. This visualization is useful to check if the graph is well sorted. If a graph is well sorted, you will expect to see a very smooth gradient, as in this case. On the bottom, we are coloring path by orientation, black path in forward and red path in reverse. Here we can see that there is a path in reverse orientation with respect to the other ones. That's the same path that have on the top here, the gradient in the opposite direction with respect to the other gradients. We can color, or we can also generate 2D visualization to visualize the graph layout. And in the graph layout, a region of variation becomes, the jargon here is that become bubbles in the graph. And to generate this kind of visualization, this kind of layouts, we apply the graph sorting algorithm that can work in 1 dimension on the top and 2 dimension on the bottom. And this is a visualization, an animation that shows how roughly the algorithm works. Okay, here is another example of a graph layout. I've reported this visualization to highlight that we usually work with the chromosome-specific pangenome graph. So we partition the assemblies by chromosome, and we make chromosome 1 pangenome graph, chromosome 2 pangenome graph, but if needed, we can you can also apply Pggb on pangenome graph representing also multiple chromosomes together. This is an example of a pangenome graph representing 5 human chromosomes. In this example, the 5 human acrocentric chromosomes. Here I'm using a tool to visualize dynamically pangenome graph that is called GFestus. Unfortunately, this is not a well-maintained tool, and it's also hard to install. But if someone will be able to install these tools all locally on their machine, would be nice also to use it to explore the path, the graph that we will generate during the hackathon next week. We usually generate chromosome-specific pangenome graphs representing the whole chromosomes. Pggb is a very, I would say, general and simple approach. We take all the sequences without removing any regions, and we create a pangenome graph. So we usually don't filter out centromeres. On the right, this is a pangenome graph of human chromosome 6 with the 90 haplotypes. On the right, this is this complex rectangle region is the centromeric region of chromosome 6. So we usually generate whole chromosome pangenome graphs, but usually people is interested also in analyzing specific locus. You can extract with Odgi extract loci of interest from your pangenome graphs. For example, we can extract from this chromosome the complement component 4 locus that appear with this structure. This structure is very similar to what in Italy we call a caciocavallo. Click the link if you're interested in understanding what a caciocavallo is. But joke aside, this structure has a particular meaning. It reflects the biology in this locus. Because this locus exists as 2 genes, C4a and C4b, that can be present in multiple copies in this locus. And this is represented with this bigger loop. So paths go multiple times in that loop to represent I have multiple copies of these genes. But these genes can be present in 2 forms, short form and long form. And the difference is in the short form, this retroviral sequence is absent. Short form, sorry, here. Instead, in the long form, this retroviral sequence is present. And this is reflected in the graph layout with this nested little loop. So when the sequences have this retroviral sequence, they will go also through this loop. But to better have an overview of what's happening inside this locus, we can also untangle this graph with Odgi untangle. With the untangle, we can take these relationships that are represented as a loop in the graph, and we can linearize these relationships between your path in the graph and with respect to some other path. In this example, I put inside the pangenome graph the annotations taken from the GRCH38. I took the annotation from for C4a, C4b, and the retroviral sequences. You can see them on the bottom of the plot to the right. How to interpret this plot? Let's take an example. For example, these 3 haplotypes here on the left, there are the names of the haplotypes. You have 90 haplotypes here. This is a HPRC pangenome graph. So about these 3 these haplotypes, this representation tell us that these 3 haplotypes, they are the same. So they go through this loop. In the first round, this purple arrow highlight that the first copy of the C4 genes, the first copy is in long form. So it has the retroviral sequence. So as I told you, in the variation graph, the paths are represented as walks through the graph nodes. So let's go through the nodes. The first copy has the retroviral sequence, so it goes through this loop. Sorry, can you see my pointer? I'm using the pointer to go through the graph. Can you see my pointer on the slide?
Yeah.
Okay, thanks. So we go through this graph. We have the retroviral sequence in the first copy. So we go through this little loop. We move further. The arrow is orange. Orange says this is a C4A gene. We move it further. And then we have a second other arrows here, the second set of arrows here. So it means that these three haplotypes go through these big loops twice. So we have to go again inside this loop, but this round, we are missing the medium-sized arrow. This is tell us that the second copy of this gene in these three haplotypes is in short form. So here, this copy is missing the retroviral sequence. So in the second round, we go in this way, we skip this little loop, and we move further. We will generate all these steps, graph extraction, graph untangling, and this locus also during the workshop. So that's where a few examples on how we can also visualize and practically work with the pangenome graphs. And that's it from my side. So any questions?
Thank you, Andrea, for a wonderful presentation. I don't know if the attendees have any questions for Andrea. Any questions? Oh, yes, Aitan.
I can see the chat now.
Hello, can you hear me?
Yes.
Okay, thank you. I think this is a question for either of the speakers. Thank you, very interesting. It's a it's a lot to wrap your mind around, and it's a different way of thinking of things. Coming from my background of just SNPs and linear genomes, when you look at these, especially the trios that have been done, sort of graph genomes of them, are these loops and bubbles? Does one see that they are either inherited from the one parent or the other parent? Is that generally the pattern that one sees, the same as small sequence variations?
I never generated pangenome graph from trio, but the jargon, as I said, variants become bubbles. So the variant that you see the inheritance in the trio, it will represented as bubbles. So you will see the paternal go through a branch in the bottom of the bubble, the maternal to another branch, the son or daughter to one of the two branches. So you will see this kind of things.
Okay, thank you. Yeah, that was my question.
Yes, Chris, you can ask a question.
Yeah, so I think when we did this in the workshop, I think we used phased assemblies, right? Or it might have been maybe some phased assemblies, some non-phased. So we had the maternal and paternal assemblies from Hi-Fi ASM. Is that correct?
Sorry, in the example that I've shown you, it's always in the HPRC we used Hi-Fi based assembly plus trio data, so they are almost fully phased. Almost fully phased. Okay. So the quality of the assembly obviously will play a role in whether or not you want to use fully phased. So the trio assembly obviously is going to have better resolution than, say, a Hi-Fi only assembly. Yes, having a phased assembly is also nice because you can also think about, I have two variants, two bubbles, they are in cis or in trans. You can also think about this kind of analysis that you can't do faithfully if you don't have phasing information. Okay, thank you.
Are there any other questions from the attendees that you'd like to ask Andrea regarding his presentation? Okay, it looks like we don't have any questions? Thank you, Andrea and Glenn, for such wonderful presentations. And hopefully the attendees benefited quite a lot. I'd like to make an announcement regarding tomorrow's presentations or tomorrow's talks. They will start at 4 o'clock. So see you tomorrow.
Bye-bye.